Okay, colleagues, it's 12:05. I would like to start the presentation. Colleagues, please join me welcoming Dr. Fortunate Mokoena, who is very energetic researcher in Northwest University from South Africa. She leads research in infectious disease drug discovery. Her work focuses on developing novel therapeutics for diseases that disproportionately affect children under 5, particularly malaria and pneumonia. She has published in internationally peer-reviewed journals, presented at major global conferences, and received funding from Grand Challenges Africa and South Africa's TIA, Technology Innovation and Agency, to advance her research program. Dr. Mokoena is a full member of the Grand Challenge Drug Discovery Accelerator and lead the drug discovery team for the Afrocentric Disease Core under the National Institute for Theoretical and Computational Sciences Initiative. Colleagues, please join me welcoming Dr. Fortunate Mokoena. Dr. Mokoena, we are very glad to have you, and we are looking forward to have your exciting results sharing with us. And we are also very grateful to you for accepting the invitation and giving the talk. Dr. Fortunate Mokoena, the floor is yours. Thank you so much.
Well, thank you very much, Prof. Syed, and thank you so much to the head of Biochemistry and Microbiology at the University of Zululand for the privilege to really share our work and perhaps maybe give a bit of highlights of the story that is busy developing out here at the Northwest University. It's my pleasure this afternoon to talk to you about democratizing drug discovery in Africa, accelerating malaria therapeutics through artificial intelligence. All of a sudden, I'm not able to move my slides.
Okay, you can unshare and share it again, please.
Okay, let's try that. Okay, so I bring my greetings really from the Mafikeng campus of the Northwest University. We are divided into 3 main campuses. It's the Vanderbijl as well as the Potchefstroom, but our biochemistry department is based at the Mafikeng campus. It is one of the 2 biochemistry departments at the Northwest University, with another one at the Potchefstroom campus. And most of my talk will be focusing on work that was done by present and former members of the Parasite Drug Discovery, which is my lab here at the Northwest University. Some of the work was done by Khalifon Tsiani, who's a former member of our lab, and Tato Masodi, who's also a former member of our lab. And really 2 members who are championing the work and are heroic, really performing some of the major experiments in the lab right now, December Patrick Makatsela, as well as Khajamohiddin Maloka. I don't know. Okay. So since the focus of my talk is really about malaria, I thought it would be useful to talk about how malaria still remains a challenge. It really is a pandemic. And in 2024, WHO indicated that malaria was responsible for about 610 deaths all over Africa, and about 95% of these deaths happen in sub-Saharan Africa. And the disease of malaria is disproportionate in the fact that it affects high-risk population and very much vulnerable population, which is children under the age of 5 and pregnant women. And just to take you through a little bit about how the malaria infection works, is that what happens is that you've got an infected Anopheles mosquito. So the mosquito would be infected by the causative agent of the disease, and the causative agent of the disease is Plasmodium species. So when an infected mosquito bites a human, when it tries to get a blood meal, it would deposit motile forms of the parasites, which are called sporozoids. These sporozoids, once they've entered the human host, they will migrate to the liver. When they arrive at the liver, they begin to divide and proliferate. And thousands of these the sporozoid convert into merozoid, and thousands of these merozoids then rupture out of the liver and begin to invade the blood. Once they enter to the blood stage, they begin to convert into various forms of the parasite, and this process of converting into various forms of the parasite results in abrupt temperature changes in human, causing really the disease manifestations of malaria. So once the parasite enters the blood stage, the merozoite would be converted into rings. From the rings, they would be converted in trophozoites, and from trophozoites, they'd be converted into schizones. Schizones have got one of two possibilities. They can be taken up by another mosquito to continue the infections of malaria in another human being, or they can proliferate into the sexual stage, which is characterized by gametocytes, continuing the cycle of malaria if no treatment is administered. So at the asexual blood stage, that's where we see the disease manifestations to malaria disease, and so treatment options need to concentrate on this as a strategy to manage the disease that would already be occurring in the particular host. The second approach, perhaps maybe to malaria treatment, would be to try and treat the parasites that are in the liver before they begin to spread and cause disease. So this would be a prophylactic strategy, or you could also try and treat transmission here. So perhaps maybe trying to block the mosquito from depositing sporozoite into the human. So these are the main strategies that have been looked at all over the world around malaria treatment and therapeutics. But the current frontline treatment option for malaria is administering combination therapy, and how this works really is that you've got a combination of two drugs. This would be an administering derivatives, and in this illustration, this would be an artemether. So this is fast acting and it's attacking the parasites at the blood stage. So the parasites that are already causing a disease manifestation would be attacked by this particular drug. The second one, the lumefantrine in this case, is slow acting on parasites that would probably be in the liver stage and already kind of like lying dormant, so that when they come in after 7 to 10 days, they could still be able to find drug that can effectively get them out of the system. So this is really the main strategy that has been looked at in terms of malaria treatment. Unfortunately, we're beginning to see cases of drug resistance in malaria, and the first case was reported in the east of Asia, in Cambodia, where really slower clearance of the parasites using this artemether combination was beginning to be recorded. Unfortunately, these cases of resistance have begun to spread, and they're becoming a lot more common, with cases being reported in Rwanda as well as in Uganda. So this risk of drug resistance strain really indicates to us the urgency of needing to find new therapeutic options to treat the disease of malaria. And in consideration perhaps of future anti-malarials, we need to think about drugs that are multi-stage targeting. So potentially drugs that are looking at transmission blocking, that are looking at treatment in terms of looking at the asexual blood stage, as well as perhaps maybe targeting the prophylaxis stage in the liver stage of the parasite. We probably need to think about a single dose cure, which means if we get a molecule that is targeting all of these stages, it can be taken by patients as a single dose cure. We need drugs that are inexpensive and well tolerated. This is because malaria is highly predominant in Africa, where we're really not, we're a poor continent. So we need to really try and think about medication that is inexpensive, that can be affordable by Africans, and it needs to be well tolerated. As I said, the disease is highly prevalent in pregnant women and children under the age of 5. So we need to consider the vulnerability of the population that the disease is prevalent in. Hopefully, we can develop molecules that have got a good resistance profile, such that we can administer these drugs for a longer period of time before cases of resistance begin to emerge, and they need to also be fast acting. So the Medicines of Malaria venture, which is really responsible for the development of new antimalarials, have given some recommendations in terms of what we could think about around future antimalarials. And the idea here is that we probably need to look at new targets. So we need to find protein targets that have not been targeted by the currently available therapeutic options, so that then the cases of resistance do not emerge as rapidly as they have emerged in the past. So the idea is to try and find novel targets with distinct mechanisms of actions, and this would be proteins probably, or drug targets that are crucial for disease progression and pathogenesis. And these proteins or the targets needs to be essential for parasite survival. So if the protein is present in the parasite, it allows for the parasite to survive, but once you inhibit the protein somehow, it should be possible for the parasite to be dead, and that would be your therapeutic strategy. And obviously the protein targets need to be validated in preclinical mouse models. And MMV has also given a criteria about what could be considered to be a hit molecule. So when you're starting in your early phase of drug discovery and you are looking at variety of compounds, which ones would you consider to be maybe having activity against malaria parasite? Is that you need to look at a compound maybe with a cellular potency, an IC50 of less than 1 micromolar. You need to have a compound potentially that has got a selectivity of greater than 10-fold against mammalian cells. So when you're looking at probably the safety profile of the compound, and you also need to look at a compound that has got an on-target engagement of a KD or an IC50 of less than 1 micromolar. So our lab really was focusing on this criteria that was given by MMV in our approach to try and develop novel anti-malarial drugs. But perhaps before I talk about the work that we've done is to think about the drug discovery process, which is really a long and odious journey. It is an expensive journey. It's been determined that it cost about $1 billion from the process of discovery to a development of a drug. But in simple terms, or maybe just to break down the discovery process, not necessarily the development in terms of the timelines that it takes, is that firstly, during your drug discovery process, you begin by looking at maybe target identification. This is when you think about your particular protein of interest and whether you can link it to a particular disease. Do you know that the protein that you are looking at is involved in the disease? Do you know that if you inhibit the protein, for example, the organism dies? That is your target validation stage. After that, you need to find then compounds that can act against your protein of interest. So that is your hit identification, which takes up to 3 years sometimes, and then hit validation, which could also take about 3 years, and then you then activate a hit to lead campaign. This is the idea of improving whatever shortfalls that you get out of your hit compounds, and this can also take up to 3 years, moving into lead optimization, where you are improving now probably mostly the drug-like properties of your compound of interest. And this process also can take up to 3 years, and then moving on to candidate selection, where you are beginning to now treat this particular compound in a preclinical setting using mice models or whatever kind of organism of interest that you have. So this is really the process of the drug discovery pipeline, and it takes long, and it's an expensive process, and it's sort of like an expensive undertaking. And our idea here that we are sort of proposing is that perhaps we can use machine learning algorithms to try and then shorten this timeline, to try and bring Africa to also play a role in the drug discovery process. So the Malaria Drug Discovery Accelerator, which is an international consortium that is working on malaria and coming up with novel drugs against malaria, have identified from the genome of P. falciparum a list of proteins that could be considered to be targets, and these proteins have been prioritized into various categories. So there are some proteins which have been deprioritized. This is probably because some of the drugs that are targeting these particular proteins have also shown some resistance profiles. There are also new and emerging targets that researchers all over the world can begin to consider. There are also some targets that are also under considerations. There's also what they have as a high priority target. So these would be often targets that are either milder or different pharmaceutical companies are looking at in terms of anti-malarial drugs that are already in the pipeline. Our interest is really this particular HSP90, which is also endorsed by the MALDA consortium as an important target to look at in terms of malaria. Just to probably maybe give us a bit of a background in terms of how HSP90 works, is that HSP90 is a molecular chaperone, and it is a structurally a homodimer, in that you've got two monomers that are exactly identical to one another. And the genome of Plasmodium falciparum encodes for 4 HSP90. So you've got PF-HSP90, and when we say PF-HSP90, we mean the HSP90 in the cytoplasm. And then you've got HSP90 that is in the endoplasmic reticulum, that we call it PF-GRP94, HSP90 that is in the mitochondria, we call it PF-TRAP, and HSP90 that is in the apicoplast, which we call PF-HSP90_A. So how HSP90 works, and in this case, we're going to be focusing on PFHSP90, the cytosolic HSP90, is that during protein folding, so when proteins are getting made and they need to be maturated into their active conformation, a partially folded protein would be recognized by HSP40 as it comes out of the ribosome, and HSP40 would take this protein and bring it to HSP70. HSP70 would then partially fold the protein, and then HSP90 in its open conformation, as I spoke about the fact that this is a homodimer with two monomers that are exactly identical to one another. In this open conformation, HSP90 would be ready to accept a partially folded substrate. So HSP70 would be assisted by the action of the HSP70, HSP90 organizing protein, which is HOP, which binds to the TPR, uses its TPR domain to bind to the EEVD motif on HSP90 to deliver this partially folded protein. Once the partially folded protein enters the HSP90 kind of like C-terminal domain, HSP90 then binds to ATP, bringing the two N-terminal domain of the HSP90s closer to one another. And the action of the PP4, which is a co-chaperone of HSP90, moves the partially folded protein slightly into the middle domain. Then the AHA1, which is another co-chaperone of HSP90, allows for the crossover of the middle domain or dimerization of the middle domain, therefore providing the requisite energy required for fully maturating the partially folded proteins into its mature conformation. This is how HSP90 really folds proteins that are coming out of the cytoplasm and need to be converted into their overall three-dimensional form. So because of this role that HSP90 plays in proteostasis or protein homeostasis, it is not surprising that it is required for the survival of many organisms. In fact, in eukaryotes, about 400 client proteins of HSP90 have been described. So in this perspective, HSP90 has been found to be critical for parasite survival. So our reason for believing that HSP90 is a great drug target for malaria treatment is that PFHSP90, the cytosolic one, has been implicated in drug resistance. And this was shown really that there is a direct interaction between the chloroquine-resistant transporter protein. So the protein that is really responsible for conferring resistance to chloroquine is a client of HSP90. But it was also shown in the same study that if you take a drug-resistant parasite and you treat them with chloroquine and an inhibitor of HSP90, you can reverse that resistance mechanism. HSP90 has also been identified as one of the 15 administration combination therapy resistance determinant. This was a treatment study where they put through administration combination and grew the parasites until they developed resistance, identifying about 15 genes that could potentially be responsible for resistance mechanisms, and one of them was HSP90. It was also shown that, once again, as I've indicated, that PFHSP90 inhibition could circumvent resistance mechanisms. In a recent study, it's been shown that inhibition of PFHSP90 has a high bar of resistance. And effectively what happened here is that parasites were treated with PFHSP90 inhibitors, and they were subjected to growth and patch surges for almost 1 year, 8 months, and no resistance emerged. So we believe that targeting PFHSP90 could circumvent resistance mechanism, or it would take long before molecules that are targeting PFHSP90 could develop a resistance. Another reason why we think PFHSP90 is an impressive drug target is that it is validated for malaria. This was shown in a study where it was shown that PFHSP90 is crucial for disease progression pathogenesis. This was patients who were divided into 3 groups. Some of them were given high fat, high glucose diet. Some of them were starved. Some of them just presented with the disease. And in all scenarios, you could see that HSP90 expressions were upregulated, indicating its crucial role in the pathophysiology of the disease. Studies have also shown that PFHSP90 inhibition is critical to the liver stage, to the asexual blood stage, and to the gametocyte, which means that molecules that are targeting PFHSP90 could be administered as a single dose cure, and they could be targeting multiple stages of the parasite life cycle. PFHSP90 is essential for the survival of the parasite and was validated in preclinical models. So pretty much what happens is that different studies have shown that if you take parasite mice models and you infected them with P-Bug and treated with PFHSP90 inhibitors, you could effectively cure the disease. So that is the level of validation. So we think that this work provides compelling evidence that PFHSP90 is an impressive drug target in malaria. But of course, despite how great it is as a drug target, there is a great challenge with looking at chaperones, and this is because molecular chaperones are highly conserved across different organisms. So if we look at the fact that perhaps maybe, despite its great or promising as a drug target, it is possible that due to their conservative nature, as you are targeting the PFHSP90, you may also unintentionally affect the human HSP90. We then looked at the sequence identity, particularly focusing on the N-terminal domain of PFHSP90. So if you look at the N-terminal domain of PFHSP90 comparing to human HSP90 alpha and beta, you can see that there's very much strong sequence conservation with only 3 main substitutions at the ATP binding region. Right? So this obviously is the reason why there are challenges, maybe raising the likelihood that there could be a potential cross-reactivity between the human and the PF. But once again, if you look at this is crystal structures of both the human HSP90 and the PFHSP90. If you look at the human HSP90 engagement with ATP or ADP here, which is a natural ligand of both proteins, you can see that for human HSP90, this engagement needs to be mediated by magnesium. But in the case of PFHSP90, magnesium is not required for ATP hydrolysis. And here you can see that arginine-98 in PFHSP90 seems to be the one that is coordinating the hydrolysis of ATP, which means that the 2 chaperones are acting in a different mechanism of action. And the presence of this arginine-98, which is a different amino acid in human, suggests that we could exploit interactions with arginine-98 to drive selectivity. So we once again looked more closely in my lab in terms of like, could we engineer molecules that are selectively targeting PFHSP90, once again by exploiting the arginine-98. And here you could look in the pink here, it's human HSP90. So it's human HSP90 in different conformation. This is the open state and this is the closed state. So if you look here, you've got arginine-98, which is here in about pink or so, and then you've got the lysine. You can see that these 2 residues are adopting different conformations, even though they are protruding inward into the ATP binding pocket of the 2 chaperones. Here in another conformation, actually the lysine flaps out the human equivalent form of this arginine-98. So we think that obviously based on this planar orientation of the arginine-98, we can then use computer-aided tools to be able to design molecules that are selectively targeting the PFHSP90 and then obviously disrupt this shorter distance that is found in the human HSP90. Another way we can look at selectively targeting the PFHSP90 is that if we look at the glycine-rich pocket here, in PFHSP90, this seems to be a straight conformation. Meanwhile, in the human counterpart, this is a curved conformation. So we can really engineer molecules to exploit this larger pocket to introduce some hydrophobicity in order for us to really selectively then inhibit the PFHSP90 without affecting the human HSP90. I hope that this illustration has demonstrated to you that perhaps it's possible to engineer selectivity using computer-aided approaches. And this is what we've done. We've published this work in Transaction of Social Societies. Please look at the work. So if we follow once again the criteria that I've spoken about from MMV, is that you would then think about a hit compound being any molecule that exhibit a potency of less than 1 micromolar and a selectivity index of about greater than 10. And this was really the criteria that we were following in the 2 studies that I'm going to talk to you about right now. In the first study that was driven by Tato Matlodi, we looked at AutoQSA-driven active learning. So this is basically teaching machine learning models over and over how to perform maximally for you to be able to prioritize compounds that could, in this case, be inhibiting or acting towards the PFHSP90. So what we did was that we looked at the literature and we found a paper by Everson et al. And then in that paper, we found 2 compounds that were mirroring the activity of what is a candidate hit compounds by MMV criteria. So we had compound 7 and compound 10. Once we got these 2 compounds, we then performed induced fit docking. to see which of these two compounds had a better fitting into the ATP binding pocket of PFHSP90. And we saw that compound 10, even though it had slightly lower potency compared to compound 7, had a much better fitting into the ATP binding pocket of PFHSP90. We could see that also compound 10 was forming these packet ion interactions that we could potentially exploit for a reaction-based enumeration reaction that followed. So we chose then compound 10 to be our reference compound. We took this compound and we subjected it to pathfinder analysis in order for us to retrospectively understand the synthetic pathway and then break down a core in order for us to introduce new ideas using machine learning algorithms. After this, then we performed standard docking. This is where we were prioritizing the ideas that were coming out of Pathfinder. We also included molecules from the Campbell database in order to enrich the chemical space. Once we'd done this, we would take 1,000 ideas from our enumerated products, 1,000 ideas from Campbell database, and then we would build the machine learning model. We continuously did this process until the learning model reached convergence. And convergence means that the statistical parameters of the model that we were looking at were no longer improving. After this, then we looked at whatever compounds were prioritized. We looked for their ADME properties, which is absorption, distribution, metabolism, excretion, and toxicity profile. So this is an in silico prediction of these particular properties in the molecules that we came up with. After this, then we conducted induced fit docking, which is a more robust and more exhaustive docking search. And then we did molecular dynamic simulation to be able to really see if we can get a sense of what happens when you've got a solvation model into the system. And then from then onwards, we predicted or we calculated the free energy calculations using MMGBSA. Just to basically indicate what I've been talking about is that our acting model sort of was able to predict ideas with docking scores that were mirroring the reference compound. So how we then came into the pathways that we used, obviously Pathfinder, and then we were able to see that we could break down this particular core, remain with this core, and break down this group, and then introduce new ideas. Once we did that, we built the first model here. If you look at the first model here, these are your statistical parameters. The overall score needs to be closer to 1. Right? And all of these other ones are also needing to be closer to 1, except for standard error here. So in the first set of ideas that we have here, you can see that we've got compounds clustering in this region, all right? These, because of how they are clustering in this region, this means these are chemically similar compounds. But our highly active compounds is this one and that one. So these are the two reference compounds that we were using. So even though our model was sort of accurate, it was not able to predict with the highest of accuracy compounds within our high active area. Once we got to model 14, which is our convergent model, you can see that compounds are beginning to populate here within our high active region, which means that the machine learning model had traversed the chemical space and was able to predict compounds that could match the affinity of our reference compound. We prioritized some of these ideas for patches and evaluated them against for biological activity. So this is some of the data that we got when we prioritized about 20 compounds. We partnered up with H3D at the University of Cape Town, and we were able to look at the anti-plasmodium activities of the compounds. This is against the drug-sensitive strain of Plasmodium, and I illustrate with this robot. Red is bad, orange is maybe, and green is great and good to go. If you look at the data that we have here, based on the criteria that MMV has around what a hit compound is, you can see that we've got a couple of promising compounds, but not really the activity that is required by MMV. But we've got compound FNT T5 here, which seems to be closer to the 1 micromolar here. Which is sort of like also closer to our reference compound, showing that our machine learning model was really robust in coming up with compounds that could recapitulate the activity of the reference compound. This is the activity of hamine, which is a known inhibitor of PFHSP90 and a highly potent one. We also evaluated the activity of the compounds against mammalian cells. So this is for us to be able to get a safety profile. If you remember the illustration that I gave from MMV, a safety profile of greater than 10 would be considered to be fairly safe. So our most active compound here seems to be fairly safe. We then also looked at its solubility profile. This is when you are thinking about the molecule being considered, for example, as a drug target, what would be its distribution pattern? And here we can see that the compound seems to have some moderate solubility. So here, this data shows that compound FNT5 seems to be a promising candidate hit compound for malaria drug discovery. It has got some selectivity, and it is also a moderately sort of like a distributed kind of like oral indication type of drug in the future. We also looked at now whether the compounds, despite the fact that we could show the biological activity, biochemically, can we say that the compounds are interacting with PFHSP90? Do they have some form of a binding towards PFHSP90? And we measured this using dissociation constants using SPR. If you look at the data here, you can see that the compound seems to have a very high binding affinity, which means that this antiplasmodium activity that is impressive here versus this 19 micromolar can be explained by the fact that the compound is also interacting with other proteins within the parasite, raising the risk potentially of side effects. But I hope that with this illustration, I've demonstrated to you that AL models enriched by docking is capable of traversing the chemical space, that active learning models can predict inhibitors of PFHSP90, and it was able to generate two subsets of compounds. These are adenine moiety-containing compound and pyrimidine moiety-containing compounds with these parameters. We have published this work in +1, and you are please look at it. The second approach that we looked at was pharmacophore-based hypothesis. And in this strategy, what we did is that we looked through the literature and found a list of inhibitors of PFHSP90 with variable potencies, variable selectivity, variable everything. So if you look here for compound 5E, for example, which was we got from the Wang study, this has impressive antiplasmodium activity around 70 nanomolar. It's got a moderately sort of okay selectivity towards the mammalian cell, and then you kind of like moved in that way where you are categorizing highly active, moderate, and then inactive. Once we got this subset of compounds, we built a pharmacophore model, and based on that characterization, the pharmacophore hypothesis said to us, of the compounds that it looked at as potential inhibitors of PFHSP90, the general feature is that the compounds are hydrophobic. They have two hydrophobic groups. They've got two aromatic rings, and they've got a hydrogen bond donor, right? This is the sort of overlay of the hypothesis. We overlaid this against our most active compound, which is compound 5E. You can see that you are getting perfect overlay, which means that the pharmacophore hypothesis was able to really predict highly active inhibitors of PFHSP90. If we look at this versus the least active compounds, we can see a misalignment, which once again validate that the pharmacophore hypothesis that we came up with could potentially accurately predict inhibitors of PFHSP90. And then we validated this using an ROC curve. Using the pharmacophore hypothesis, then we scanned through NMN and MMV database, which was about 2.6 million compounds. And then we got 34,000 initial hits. These hits were then subjected to a standard precision docking. We came down to about 34,000 ideas. We then added an extra precision filter, and then we got about 340 promising hits. We subjected these 340 promising hits towards the human HSP90, and then we filtered against 20 candidates, which could potentially be selective. This is just an illustration of that fact that the compounds that we selected could fit into the ATP binding pocket of PFHSP90. When we look at the biochemical data, we could show that most of the compounds that we predicted really could disrupt the function of PFHSP90. This is a competition assay where we're looking at the capacity of the compounds to displace geldenomycin, which is a known inhibitor of PFHSP90. So this is really represented as percentage bound FDA, which the positive control is 100% bound, which means any lower signal is representative of strong inhibition. So we can see that our compounds here are inhibiting, sometimes even better than known inhibitors of PFHSP90, which is 17AAG. And we can see various patterns of inhibition in that our compounds don't seem to be inhibiting the human HSP90 as much. So we are seeing that we're able to get that selectivity that we originally engineered in silico. And this is the data that we got in terms of the biological activity of the compounds. I will not take you through it again, but from this data, we decided to prioritize two main compounds. We prioritize compound FM2. We also prioritize compound F12. So the work of compound F12 has been driven by Khajamohiddin, who's currently doing her master's in my lab. And really what we did here, we started off by looking at the compound. We did a 2D fingerprinting against the MQ database, and we found these starting points. We took these compounds, we looked at them in terms of antiplasmodium activity, and all of them were inactive. We looked at their selectivity against mammalian cell, and they were fairly all right. And we got the idea here that we probably need to improve the hydrophobicity and hydrophilicity profiles of the molecules, really representing once again the capability of machine learning models to transverse the chemical space as quickly as possible. Some of the major work that we've been doing, which is driven really by December Makatsela in my lab, is progressing this promising hit molecule, which is FM2, to an early lead. So really moving it across that drug discovery pipeline into seeing whether we can develop it one day as an anti-malarial drug. Once again, the evidence, because we've got the biological assay, but the evidence in terms of biochemically, whether the compound is interacting with our protein of interest, which is PFHSP90, is that FM2 does interact with PFHSP90 with a KD of about 13.5 micromolar, but it is not selective because this KD is quite high. When you compare it to a known inhibitor of PFHSP90, you can see that you're getting a KD that is much lower, which is about 1.8 micromolar, which means that if we were to develop this one day into an anti-malarial drug, we need to drastically reduce this KD of FM2. We also showed using fluorescence spectra that actually FM2 does disrupt the tertiary structure of PFHSP90. So if you look here in the blue, we've got the apostate, and then once you get FM2, which is in the red here, you are getting a sort of quenching of fluorescence, which means that the protein is, the compound is really interacting with the protein. It's not as effective as harmine, but it does actually quench a fluorescence signal. Similarly, we showed that same effect in terms of a tryptophan spectra, that FM2 does actually quench the fluorescence spectra. The secondary structure filaments of the protein seems to be quite robust, and none of the compounds seem to have disrupted any of the secondary element of the protein in a secular dichroism. But we could show really that the compound does interact with PFHSP90. This is really, once again, a repeat of the biochemical data, the biological data that we got, the potency in terms of whether the compound does have antiplasmodium activity. We looked at this and compared it to existing antimalarials. We're seeing our margins. We're seeing where we need to get to in order for us to develop this one day into an antimalarial drug. I won't spend a lot of time discussing most of this data, as I've already explained it. The flag here is the binding affinity. We need to take this down to about 10 nanomolar. We also need to see that as we are improving the binding affinity towards PFHSP90, the selectivity is maintained towards the human HSP90. The other issue here is that our compounds seem to be moderately metabolically stable. So metabolic stability indicates the percentage of compound remaining after you've treated the compound with liver microsome. So what you do is you take the compound, you expose it to liver microsome, and after 30 minutes, you watch for the percentage of compound remaining. If we look at human liver microsome, we see about 86% of the compound is remaining. But in rat and mice, we're about 41% of the compound is remaining, which means that if you've got lesser compound remaining, you've got lesser compound to be able to get to the site of action and exert a therapeutic effect. So we need to really also improve the metabolic stability of the compounds, and the metabolic stability is obviously related to the clearance. So if you've got 86% compound remaining, it means that 12.4% of the compound has already been cleared out of the system. It's already been excreted. So here the idea is for us to get at least 93% of the compound remaining and less than 7% of the compound cleared out of the system. So because of the fact that we couldn't really correlate the antiplasmodium activity of the FM2 compound to that of the binding affinity towards the target, we could speculate maybe that it is because PFHSP90 encodes 4 HSP90s in the endoplasmic reticulum, mitochondria, and apicoplast. So we speculated that potentially what is happening is that the compound is interacting with the ATP binding region of these other isoforms of HSP90. So in order for us to get this understanding, we conducted a sequence identity, a sequence alignment, and we could see that actually the N-terminal domain of of HSP90s in Plasmodium are less conserved. They are so less conserved that the human HSP90 seems to be better conserved compared to them. If you look at the overall sequence alignment, you can see that the residues that are involved in ATP binding seems to be reasonably conserved as opposed to this one where you are having here the arginine-98 being a lysine in the GRP94 and the TREP1, and then also a glutamic acid in the apicoplast. But the apicoplast has got an insertion here. And also the mitochondrial HSP90 also has an asparagine-rich insertion here, which suggests that our speculation perhaps that the compounds may be interacting with other isoforms of HSP90 may hold true for GRP94, but not necessarily for the apicoplast and the TREP1. In terms of structural alignments, pretty much what I've explained in the sequence identities being recapitulated. We're illustrating here that the lysine versus the arginine-98 represents an opportunity to engineer selective inhibitors. So what we then did is that we used the ligand designer module within the Schrödinger Maestro to then conduct some deep learning in order for us to enumerate and generate new ideas. And our plan here was to do a bioisosteres replacement in order for us to get a better core of this FM2 better than this one, which is not chemically tractable. I'm just going to illustrate these ideas here where we've removed the carboxylate group here and also introduce like a amide group here in order to try and see if we can get better competing molecules than the FM2. Biochemically, we could show that the compounds that we've generated using ligand designers were able to shift the melting point of PFHSP90, not so much of GRP94 and not so much of human HSP90, showing that perhaps the new ideas really could be selective. We could also show that the compounds inhibit PFHSP90 better than GRP94 and human HSP90, once again suggesting that they could be selective. We could also show using limited proteolysis that the compounds induce a better conformational stability towards PFHSP90 compared to GRP94, showing really that the compounds could be selective. We also showed here using SPR that our compounds had better binding affinity towards PFHSP90 compared to the original reference compound, which is FM2. However, the compounds lost potency because as we introduced the new ideas, somehow the substitutions were too radical and we lost the antiplasmodium activity. But I hope that what I've been able to illustrate to you this afternoon is that the ligand designer assisted generation of novel molecules produce compounds that describe PFHSP90 function more effectively than PFGRP94. The newly designed analogs induced a more stable conformational state in PFHSP90 compared to PFGRP94, and thermal shift assays showed that the molecules significantly shifted the melt curves of PFHSP90 with smaller effect on PFGRP94 and no measurable effect on human HSP90, suggesting some potential selectivity over there. The analogs also demonstrate improved binding affinity compared to the original compound, which is FM2. Although the new analogs ultimately lost potency, this finding highlights the value of AI-driven analysis for rapid decision-making and optimization in drug discovery. I'm really, really grateful to all the funders that have believed in our work and supported it over the years. We started this work with a grand challenges that was awarded by the Bill and Melinda Gates Foundation, and we're continuing it with money that we got from the South African government through the Technology Innovation Agency. I'm sincerely grateful to all the collaborators across the world who have contributed immensely towards the work that we're doing for us to get to be here. And I'm deeply, deeply grateful to members of my lab were really the hands and the feet who are doing this work. Thank you so much for the time to share our story.
Dr. Mokoena, thank you so much for sharing such an exciting research going in your laboratory. I see you are very close to get some good inhibitor. It's really, really great, great. And the way you presented your work also, it is easily understandable different steps for honor students because some of the attendants are honor students actually from clinical
Yes, I thought.
biochemistry.
Yes.
So this is very good. Also, one of your slides include drug discovery process, clearly
Yes.
showing how many years it takes
Yes.
from identification of a target to release the drug in the market. I am sure most of the honor students are now realizing how long it will take to just to get a drug in the market or identifying how many steps are involved because all most of our students they think they just identify drug, they want to cure the cancer, they want to cure the malaria. So I hope students, all of you are getting some good information. This is not a one bullet shot.
Yeah.
Also, the way you focus it on HSP90, which is no resistant even after treating a couple of drugs, is very appreciable. And one thing I like it in your slides, the steps you included for drug discovery, how your molecule take to the different processes, Pathfinder and ADME, then QSR. This is very excellent presentation, especially for clinical biochemistry students. We have one topic, drug discovery and drug is this thing perfectly suitable. Also, finally, I want to ask you, FM1, FM2, FM3, I know it's not a radio station, right? What is FM means?
Unfortunate Mokoena.
Oh, you named your compounds after you.
So the because of intellectual property, we can't put the actual name of the compounds so that one day if we need to protect that intellectual property, we're safe. So we hope that we've presented enough for people to get a sense of what's going on in terms of the structure is there. You're able to work out what functional groups are there and what not. But obviously, we work in collaboration with our technology innovation support services, and they give guidance in terms of what can and cannot be shared.
Wow, this is amazing. Dr. Mokoena, your presentation is excellent,
Thank you
both
so
at the
much.
highest level and also understanding level. Now, I will open the floor for any comments or questions. Colleagues, the floor is now open. Could you please just open your video, introduce yourself, and please ask the question. Gidaga, please go ahead. Please go ahead. You please open your video and introduce yourself and ask the question, please. Thank you. Mapumzi Gidaga, you need to unmute yourself. Yeah, we can hear you.
Can you see me?
No, we can't see you. Could you please open the... Now we can see you. Yes, please proceed.
Hello, thank you, Doc, for that presentation. I understand the swapping the carboxylic groups for the amines, because the amines will be more soluble. However, they are less hydrophobic than the carboxylic groups, yet you want hydrophobicity as well. So how do you balance now that?
Thank you very much. I think, thank you. That's a very excellent question. I think you're spot on the money. So our approach there was to, if you look at the actual structure, it's got two carboxylic groups. So that made it very complicated for chemists to make. So we were trying to make it as easy as possible. And so we had, for the metabolic stability, that is not so good in the rat and the mice. We had the suspicion of whether could it be the methoxy group on the right hand of the compound that is resulting in this lower metabolic stability, or the sulfur group on the right hand side of the molecule. So we brought in the nitrogen to try and see if maybe we could improve the metabolic stability while retaining. Obviously, the nitrogen has the capacity perhaps maybe to conduct a hydrogen bond or something while retaining some of the potency. But once again, you're spot on the money. That substitution resulted in a drastic loss of potency. So we've been able to see that you shouldn't engineer property, but maybe conservatively keep the same properties of the molecules.
Okay.
Thank you
Thanks
so
very much. I hope I've answered you.
Yes, you did.
Thanks very much.
Excellent, Dr. Mokoena. Colleagues, any other questions, any comments, colleagues? Students, do you have any questions? Please feel free. Dr. Mokoena, I see no questions. Okay, 1 minute. Dr. Mokoena, once again, I would like to thank you so much for accepting the invitation and presenting excellent research. I would like to request, is it possible, can you share 2 slides with us? One is overall drug discovery process, where you put from target identification to always the drug, how many months. The second one is active learning modeling steps, different steps you put like a pathfinder, QSR, these things. These 2 slides really helpful for our students to understand how the drug discovery process works. If possible, only these 2 slides. I know this your work has intellectual property, right? So I'm not asking any more. Just these 2 general slides. Is it okay with you?
No, absolutely. We're able to, on the AutoQuizer, we've published the work. It's in +1 2024 Mathodi. So definitely we're able to share that work.
Okay, now we have a question. Buthelezi, please open your video, introduce yourself and ask the question, please. Buthelezi, you please go ahead. Unmute yourself, please.
Okay.
Yeah.
Good day, everyone. Okay. I think I'm just interested on the first slide that you presented, of which was talking on the malaria pandemic. So like you told us like how does this malaria like, okay. So I'm just concerned about why does the human's immune system fails to eliminate the malaria once the infection begins? Because we saw that it starts when a person is bitten by the infected mosquito, then it proceeds to the liver, also the blood. So why does the immune system fails like to eliminate malaria eventually when it begins?
Okay. Thank you so much for your question. In general, humans who are healthy are able to, we call malaria infection self-limiting, okay? So a healthy human will be able to get rid of malaria, okay? But the vulnerable population for malaria is children under the age of 5. So they're still young. Their immune system and their liver has not developed as well. So the parasite then sort of overwhelm the liver. So pregnant women obviously are metabolizing a lot more than the ordinary person because they are basically doing the job of 2 people. And young kids, their liver is not at a stage where it's fully developed in order for it to get rid of some of these challenges. So, but ordinarily, humans who are healthy are able to get rid of the malaria parasite in their system without, except in the case where you are exposed to now the drug-resistant strain.
Okay, thank you. I'm answered.
Awesome. Thanks so much.
Thank you. We have one more. Please, Mr. Gidaga, please, you can proceed.
So you mentioned that you're using artemisinin derivatives, right? So with wormwood, or what do they call it, Artemisia annua, would administering or making people ingest that plant also contribute to lowering the burden of malaria in the liver? And with that being said, how, if you're looking for compounds to link to Artemether, right? Wouldn't it not be beneficial to look at the flavonoids or the terpenoids in the plant for a compound that you can link to create the molecule that you use to test?
Wonderful. Thanks so much for that sort of comment. I do think that there's value in, plant-derived molecules. The main challenge with obviously plant extracts is that you are not able to quantitatively measure the amounts of compounds that are in the extract. So because you're not quantitatively able to measure how many they are, then you can't determine dosing, which means that whether I take 2 extracts and somebody takes 3, it's the difference between who gets toxicity and who doesn't. So that's the major challenge. But just a starting point of drug discovery, if you use a plant extract and you get anti-malarial or anti-plasmodium activity, you are able to then go down to then find out what is the bioactive constituents of the actual plant extract, and then come down to then extracting, as you rightly said, the flavonoids or the alkaloids, right? The question there is how much of a yield are you going to get? So you're able to often get the activity from the entire plant, but once you go down to the fractionation, often then your yields are not enough. But what you are able to do is you also be able to get a structure maybe that can inspire medicinal chemists to design better molecules around that scaffold. But I think it's a worthwhile research. We're just looking at single molecules that are novel, that have not been seen by the parasite before, so that we can improve the likelihood that drug resistance would take us a little bit of time before it emerges.
Thank you so much, Mr. Gidaga. Okay, we have one more question. Please open your video and please introduce yourself and ask the question. Please proceed. We can't hear you. Could you please open your video?
Okay, can you hear me now?
Yes, please proceed.
Okay. No, thank you so much, Dr. Mokoena, for such a wonderful presentation, as well as the addressing of question. Mine is simple. I'm just wondering, what did you use on your plots? I see some wonderful plots on your result section. So just wondering, what plot did you use? Was it some kind of a software or
On what?
On your plots, the graph presentation of your results?
Yeah.
Yeah, I'm wondering if it's a software or just use Excel Word.
Or thank you so much, Muzomuhle, for your question. I will shout out my incredibly amazing students here, Tato Mahlodi, Hlalifontsiane, Disema Makatela, as well as Kihu. They are the ones who've really generated most of these figures, but they use predominantly GraphPad for the biochemical kind of like investigations. The other stuff is the Schrödinger software within Maestro.
Oh, okay. Thank you so much.
All right.
Thank you. Colleagues, any questions? Students, if you have any questions about the process or about malaria, please, colleagues, this is the time to ask. The floor is open for more questions. You just raise your hands and you can ask. Dr. Mokoena, so far I haven't seen. Let me say acknowledgements to you. Before that, I just want to announce students, especially clinical biochemistry honor students, you please stay online so that we can have a roll call. Okay, so please don't leave. Okay, colleagues, I think this comes to an end. Dr. Mokoena, you can switch on your video, please. Dr. Mokoena, we really thank you from the Department of Biochemistry, University of Zululand, for accepting the invitation and giving us an excellent talk with basic steps, what involved in the drug discovery process. And your presentation and the results you have in your laboratory is greatly appreciable. And congratulations to you. You have so many hit molecules. They are in the process. And we wish you all the best. And one day we want to see this realizing into drugs, a real drugs. And we hope you definitely will succeed. Once again, thank you so much, Dr. Mokoena. I'm sure we look forward to have more talks from you. Thank you so much.
So my most sincere thanks, Prof. Thank you so much for the invitation. And I'm so glad it was so engaging, and really thank you so much.
Thank you, Dr. Mokoena. Colleagues, now I will stop this recording. Colleagues, remaining colleagues,